delighted to be here. Great group of people, meeting better and better people each and every minute I'm here, so I'm definitely delighted. And what we're going to talk about today, if I can figure this out, first disclosure. All right, we have a company, so I hope you read that, so we're covered. So the company's looking at 3 clinical indications right now. We're looking at rejuvenating muscles as you age, reversing obesity-linked type 2 diabetes, and on the back burner, we're targeting glioblastoma and a number of CAFs that are involved in metastatic disease. But for today, we're going to be focused on, because it's right after lunchtime, obesity-linked type 2 diabetes. We know, you know, obesity is a big problem. Obesity is one of the leading indications for type 2 diabetes. All right, so we want to tackle this issue. So I guess my pointer's is going to work, barely. The obesity rate is pretty constant in America at 37, 38% in most age groups. When you look at the percent of obese people in age groups that have type 2 diabetes, it rises as you progressively age. So if you look at the general population, about 20% of obese people, obese being defined as a BMI over 30, about 20% have type 2 diabetes. When you're up in the age of 55 and plus, it's pushing about 1 in 2 of the obese population have type 2 diabetes. And so our first clinical indication is looking, first of all, in the elderly population, can we reverse obesity, and by doing so, obesity-linked type 2 diabetes. So that's the disease we're going for. And why are we crazy enough to do this? Because the market is flooded with diabetes drugs. $40 billion spent last year on diabetes drugs in America. There are and is very little obesity market in America. We spend a lot on treatments to reduce weight, but there's very few drugs on the market that have any success in treating obesity. So why are we going at it? Well, we think we can tackle this by a very different pathway than the current approaches. I would argue that everything on the market to tackle type 2 diabetes is treating symptoms. It's treating the symptoms of the disease. You have too much sugar in your blood, let's lower the sugar. Well, too much sugar is a symptom, okay? You're not getting enough insulin, let's add insulin. That to me is a symptom. What's the root cause? The root cause of obesity-linked type 2 diabetes is obesity, all right? And the inflammatory responses that come from that, and a whole bunch of others. So we are targeting an enzyme, NMT, it's a methyltransferase, that we believe actually will reduce the level of obesity. It directly impacts the adipose tissue, the visceral fat that occurs in obesity. So we're trying to tackle the root cause of the disease and not simply a system. So we know the structure. We've done a lot of structure-based drug design. We've done some screening. We have a number of small molecules that are targeting the methyltransferase site, not the SAM site. So this is a methyltransferase. It transfers a methyl group from the SAM to the nicotinamide. We're targeting the nicotinamide site. We can inhibit the enzyme quite nicely. Because this is an ADME talk, this is just showing a couple radar plots of just 4 parameters. Okay, we can do this on many, many different characteristics of the drug. So ideally, if I can show this. So an ideal drug would have many, many axes here in this radar plot where we're looking at many different parameters, but let's just look at 4. The IC50, how potent is the compound, would be one thing we would care about. What's its permeability? We'd like it to be a high permeable compound. What's its half-life in human, if you will, or what's its PK, some of the PK parameters in human, and how about in mouse? Okay, so an ideal compound would like to optimize all of these. That would be the ideal case. So this is one of our leads where it does pretty well, not great, but it does pretty well in the IC50, excellent in the permeability, excellent half-life in humans, no half-life in a mouse. When I mean no half-life, it is actually when you look at the oral bioavailability and how this compound performs in a mouse, it is rapidly degraded. All right? So it's not ideal. And then you can look at other compounds, and they have good and bad. This one doesn't have, it has a little bit better potency, but it's not very permeable. So you can compare these, and because it's an ADME conference, we spend a lot of time looking at a whole bunch of parameters to decide what is a good compound. One might say we try to find the optimal compound, but the optimal compound is a crazy thing to think about because there's no such thing as an optimal compound. It's like, is your spouse optimal? Well, the answer is, I won't go there, right? What does optimal mean? So the better yet is the compound good enough? I won't use that same analogy that I just did. All right. But so we've looked at a number. We like this compound because it actually is fairly potent. It has excellent PK characteristics in everything but a mouse. Okay? And these half-lives are actually based on hepatocyte studies. So when I say half-life in human, it's a half-life in a hepatocyte. It is rock solid in human hepatocytes. It's rock solid in beagles and non-human primates and rats. It is just digested immediately in a mouse. So it's not great in a mouse. And we can, working with CROs, working with Dr. Liang and the TSU group and Diane and the U of H group, number of parameters, and we saw from the hepatocytes that this compound is going to be chewed up in a mouse, and we definitely see that. If we look at the mouse PK, yes, we have nice half-life, but the bioavailability is essentially not there. So you give it orally, it gets chewed up immediately. But when you look at the rat, it's pretty good bioavailability. It's about 38%. We can do better. Half-life is pushing about 8 hours, sometimes 12 hours, things like that. So I'm about to show you that we like this compound because we think it has good oral bioavailability in everything but a mouse. About to show you data from a mouse diet-induced obese study. These are not done where we give the compound orally. We give it sub-Q, which is mimicking the IV. So we actually have pretty good compound exposure in the tissues when we do sub-Q injections in the mouse. And so that's why these work. We've tried oral or IP injections, and we see no response, which makes sense because the compound just doesn't stay around. But what we're looking at in this compound, these are diet-induced obese animals. So it's pretty simple. You take the mice, you give them a whole bunch of Big Macs continually for about 3 months. You fatten them up, and they do get fat. And they continue to gain weight for the rest of their life until very old age. We put them on treatment, shown in green, at a fairly modest dose. They either lose weight, we have studies where they lose weight, or they just don't gain weight. They eat the same amount as the control. So, analogy for the human, you can eat the same amount of Big Macs, you're not going to put on weight. Okay? You're not going to put on weight. Right. And I put on the low dose up there, so we also have some low dose studies where we don't see any change in the weight, but we'll show you some interesting results with that. So in this case now we're looking at the liver. So there's a lot of interest in what's happening in the liver in obese people and obese diabetic people. This is not a NASH model, but it's a non-alcoholic fatty liver disease model. The control groups, these are done by a clinical pathologist. The controlled mice score very poorly for liver disease. The low-dose treated groups, where you're actually not losing weight, we still see improvements in liver function. And the groups where we actually are keeping the weight low, pretty much there is no sign of liver disease at all. The livers are by and large completely clean in a few weeks of study. We can look at ketone body levels. Again, we're seeing all the doses are reducing ketone body levels, which is a sign of diabetic marker. We look at oral glucose tolerance tests. What's very interesting here, in this case for the people who do diabetes, is the green that we're looking at plasma insulin levels. Over time, the obese animals start seeing their insulin levels rise, comparable to what you see in human diabetes. our treated animals, there's no change in the insulin levels. It's processing insulin fine. Oral glucose tolerance test, we see the improvements that one would expect. And what we're seeing here is in oral glucose tolerance, this is the background level. You give a bolus of glucose, of sugar. In a diabetic patient, you see, or a pre-diabetic patient, there's a big influx of insulin as they try to process the glucose properly. In our treated animals, you don't need much insulin. You just need the same amount of background insulin to handle the bolus of glucose. So these treated animals are essentially losing weight, losing fat. All their diabetic parameters are essentially returning to normal. So we're pretty happy with this compound. Lots of collaborators, the TSU group we want to thank. And so we're moving this now into clinical trials that are targeted about a year from now. So we're finishing up all the IND studies.